Hello, everyone. Thank you very much, conference organizing committee, for accepting an approval of my abstract and offering me opportunity to share my knowledge, experience, and the research in this beautiful conference. This conference entitled "Human and Technology: A Holistic and Symbiotic Approach to Sustainable Development" is hosted and organized by the MES College, Marampally, Kerala, India. Okay, well, I appreciate Professor Dr. Manzoor, the chairman of this conference, who have worked hard to make this conference fruitfully conclusive. I'm also informed that Professor Dr. Yasmin, she became the new principal of this college. I welcome and I congratulate you for getting this new position. You have very beautiful team, including Professor Dr. Jaseena, then Dr. Rubina, then Dr. Paul, Dr. Nisha. They are very competent people and they work hard to make this event fruitfully conclusive and successful. Okay, well, my name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery Technology Development Incorporation, Ontario, Canada. I'm also working with the Advanced Multiple Incorporation, that is Knowledge Processing and Information Management Organization located in Mississauga, Ontario, Canada. Okay, well, I'm going to share my knowledge, my experience, and my research under the title of Emerging Immunomodulation Technologies that may potentially improve the clinical healthcare system all around the world. For this purpose, I collected the scientific information from different data sources like PubMed, Medline, Scoopers, American Chemical Society, Google Scholar, then MBAS, directly of Open Access Journal, ProQuest CNKI, that is China-based database company, stand for the China National Knowledge Infrastructure. Some information are collected from the Crossref and the ResearchGate. Okay, well, before moving forward, I would like to talk a little bit about antibodies. An antibody or immunoglobulin produced by a single clone cell called as monoclonal antibody. It is Y-shaped protein produced by the B cell, B means Bursa cell or plasma cell. The immune system identify and neutralize the foreign body. Foreign body may be the virus, maybe the protein, or that may be the bacteria, or it may be the any foreign objective, toxic material, or the like any kind of the challenging material that invaded or entered into the human body, that is considered as antigen. Antibody are secreted by a type of the white blood cells called as plasma cells. Monoclonal antibody or antibodies are identical because they produced from single parent cell. Polyclonal antibodies are derived from different cell line and different in amino acid sequence. Okay, well, now I would like to talk a little bit about history of monoclonal antibodies, the history of development, and their availability in clinical setting in hospitals. In 1964, Littlefield developed a way to isolate hybrid cell from two parent cell line using the hypoxanthine aminoterine thymidine selected media. In 1975, Cooler and Milestone provided the most outstanding proof of clonal selection theory by fusing normal and malignant cell. Then the Nobel Prize in Physiology and or Medicine in 1984 was granted jointly to Neil K. Gerini, George J. F. Cooler, and Cesar Milestone for theories concerning the specificity in development and control of immune system and discovery the principle for the production of the monoclonal antibodies. Okay, well, these are the three different scientists who work hard to contribute into the monoclonal antibodies. Cesar Milestone, Nobel Prize. George J.F. Cooler is also Nobel Prize. Then Littlefield Bard, he also worked hard and recognized by different institutions and organizations around the world. Okay, well, there are the three main type of the monoclonal antibodies. Whenever we classify anything, we have to decide some criteria. It may be the criteria based on chemistry, based on the functionality, or based on the source of some particular object or the entity. Here we classify the monoclonal antibodies on behalf of their chemistry and on behalf of their source. So the first type of the monoclonal antibodies is murine source, that are rodent monoclonal antibodies with excellent affinity and specificity. generated using conventional hybridoma technology. Clinical efficacy comprised of the HAMA stand for the human anti-murine antibody response, which lead to allergic or immune complex hypersensitivity. The second type of the monoclonal antibodies is chimeric. The chimers combine the human constant region with the intact rodent variable region, affinity and specificity unchanged. It also causes a human anti-chimeric antibody response. Okay, well, the third type of the monoclonal antibody is humanized, contain only the complementary determining the region of the rodent variable region grafted onto the human variable region framework. Okay, well, this is the very simple regular overview of the cell and antibody mediated immunity. We all know that the immunity comprised of the two main types. Immunity can be divided into the two main type or two main part. The first one is the antibody mediated immunity. The second one is the cell mediated immunity. We know in antibody mediated immunity there is involvement of the antibody that attack on the antigen, kill and destroy it. In cell mediated immunity, certain cytotoxic material, cytokines produced inside the body, they attack on the cancerous cells or affected cell and destroy and damage the cell successfully, that is eventually excreted from the body. What kind of the cytokine produced in the cytotoxic cell mediated immunity? There are it may be the interleukin, maybe the monokines or chymokines or lymphokines, interferon and colony stimulating factors. These are the chemicals produced in the cell mediated immunity to destroy the antigen. Antigen means maybe the bacteria, maybe the protein, or maybe the viruses. Okay, well, now I would like to talk a little bit in detail about this antibody mediated immunity. Whenever the antigen, virus, or maybe the protein, or maybe the bacteria enter into the body, they are captured by the APC. What is the APC? APC is antigen presenting cell. Antigen presenting cell may be the macrophage, maybe the dendrite cell. It engulf the antigen, process it, and convert into the epitope. Then epitope presented to the T helper cell through the through its particular protein, that is major histocompatibility complex. Major histocompatibility complex is located at the surface of APC. This is the particular place where it processes the antigen and convert it into the epitope and then put it at this particular major histocompatibility complex. It is two main type, MHC1 and MHC2. MHC1 is that is specific for the non-viral entities, and MHC that is specified for the viruses. So, like at epitope are presented to the T helper cell. T helper cell accept it, recognize it. When it understand and conceive it, then T helper cell are activated. They start producing the interleukin. Three main kind of the interleukin synthesized by the activated T cells: interleukin 2, interleukin 4, and 5. Interleukin 2 that activate the cytotoxic cells, and they start producing the cytokine. Cytokine, as I told earlier, it may be the lymphokine, maybe the cytokine, maybe the monokines, it may be the interferon or colony stimulating factors and some other chemicals produced by the cytotoxic cells, and they destroy and damage the affected cell. On other hand, the antibody mediated immunity, interleukin 4 and 5, they activate the beta cells. B cell, beta cell converted into the two main type of the beta cell, plasma cell and memory beta cell or memory B cells. Memory B cell actually store and reserve the information. If the same antigen attack or come again in future, then system activated and start producing antibody right away. And the plasma cell, this cell actually responsible to produce or synthesize the antibody against particular antigen. Okay, well, this is the very simple regular diagram of antibody. It comprises of the two main chain, light chain and heavy chain. They are attached together by the disulfide bond. The antibody having two main part, the variable part and constant part, or variable region and constant region. The constant region actually maintain the structure of the antibody, and it keep it integral form. The variable component or the variable region of antibody actually is responsible to attach with the antigen. It is variable. Why it is variable? Because it have to cover all different kind of the variety of the antigens, variety of the bacteria, variety of the viruses. So God granted him a variety of the chemistry, variety of configuration, variety of the shape. It can be changed all the time to attach and kill the all different kind of the antigen, bacteria, viruses, and other chemical entities. Okay, well, the structure of the antibody molecule, Fab domain, mediate the binding of the immunoglobulin to their cognate antigen. Each chain in the Fab stand for the fragment antigen binding domain, has variable and constant region. Variable region contain the hypervariable or the complementarity determining region, CDRs. Antigen contact residue reside in the CDR. The heavy and light chain variable contain 3 CDR each. All 6 CDR contribute or combine to form the antigen binding pockets. The CDR3 play a dominant role. FC means the fragment crystalline domain, define the different isotype of immunoglobulin. The FC fragments specify other biological activity of the molecule and determine how an antigen mediate its effector functions. Okay, well, monoclonal antibody synthesis by a particular method. The antigen is injected to the mouse or the any trial animal. The mouse become immunized. When it's immunized, it start producing the antibody. So we isolate the immunized cell like the plasma cell, and then these antibody producing plasma cells are fused with the tumor cell to produce a hybridoma. These hybridomas screened for the production of desired antibody, and these hybridoma then segregated, they are refined, they are purified, and the clonal synthesis or clonal expansion is that is encouraged to produce desired particular monoclonal antibodies. These antibodies that belong to particular antigen. So this particular antigen actually able to attach and destroy the antigen for which it is produced. So this actually this monoclonal antibody is only able to destroy and the kill that is initially injected to the mouse. So all different kind of the antigen having their own particular monoclonal antibodies. Okay, well, now I would like to uh, explain the same process in a different pattern. Like this is a mouse. Mouse is given the antigen. She is injected with the antigen, and antigen activate the immune system. In immune system, the antibody are produced by the beta cell or Barca cells. The spleen cell they fused with the myeloma cell to produce a hybridoma. These hybridoma are then cultured in hat medium selection for positive cell, and these cells are then segregated and harvested monoclonal antibodies. These synthesis monoclonal antibodies are further refined and purified and converted into the particular dosage form for the clinical or hospital use for different kind of the patients. Okay, well, this is another diagram that highlight the synthesis of the monoclonal antibodies. Antigen injected to the mouse, she is activated, and it's activated beta cell isolated. They fused with the myeloma cell to produce a hybridoma. They are cultured in the hat medium. Hybridoma then are then screened, and their competency for the production of the antibody is enhanced. Select hybridoma producing antibody of interest. These antibodies producing hybridoma are then cloned. Then monoclonal antibodies are expanded, and they are eventually packed into the particular dosage form for the actual clinical use. But okay, this is the 7-step highlighting the mechanism by which the cancer cells are killed by using the monoclonal antibodies. Initially, we release of the cell antigen, then cell antigen presented to the antigen presenting cell. The third step is priming and activation, then trafficking of the T cell to the tumors, And at the fifth stage, infiltration of the T cell into the tumor cell. And at the sixth phase, the recognition of the cancer cells by the T cell, and eventually the cancer cells are killed and destroyed by the monoclonal antibodies. Okay, this actually highlight the 7 different usage of monoclonal antibodies in different industry. The first one is the glycomodified monoclonal antibodies, then altered amino acid in constant region. The third one is the use different human monoclonal antibody isotope. Then it can be linked with isotope to monoclonal antibody with a stable linker. Fifth one is link drug to the monoclonal antibodies with cleavable linker to deliver it in different spot inside the body. Insert the DNA for monoclonal antibodies variable region fused to signaling peptide into the T cell to include expression of the CAR. CAR stand for the chimeric antibody receptor. The seventh one, the cross-linking region from two monoclonal antibodies. Okay, there are the 5 different advantages of monoclonal antibody. First one is the homogeneity. Monoclonal antibodies represent a single antibody molecule that bind to antigen with the same affinity and promote the same effector functions. Second one is the specificity. The product of single hybridoma react with the same epitope on the antigen. The third one is immunizing antigen need not to cure or characterize and is ultimately not needed to produce large quantity of antibody. Fourth one is the selection. It is possible to select for specific epitope specificity and generate antibodies against a wider range of antigen determinants. The fifth one is the antibody production. Unlimited antibodies of the single well-defined monospecific regions. There are also certain disadvantages of monoclonal antibodies. First one is the affinity. Average affinity of monoclonal antibodies are generally lower than polyclonal antibodies. The second one is the effector functions. Because antibody is monoclonal, it may not produce the desired biological response. Then specificity. For monoclonal antibodies against conformational epitope on native protein may lose selectivity and antigenicity. Okay, well, the fourth one is the cross reaction or cross reactivity. Antibodies sometimes display unexpected cross reaction with unrelated antigen. Time and effort commitment. So a long time and very long effort is needed to make this protocol or this scheme successful in clinical medical practice. Okay, well, in conclusion, there are technical difficulties in developing the monoclonal antibodies drug. However, the domestic and international industries have sufficient scientific collaboration to design, produce the accurate monoclonal antibodies. The biotechnology advancement and investment in the research continue the development of the novel monoclonal antibody drug in China and all other countries, and they will enter into a new era in next few years. Okay, well, before closing, I would like to salutate and greet my colleague Sherry Bajkat, Wolfgang Dang, then Daniel, Justin Hacker, Marwa Al Maghribi, Terry Mill, then Faisal Nureen, Kunza, Robert Heyman, Amin, Arslan, and Ishtiaq. They are my people. They helped me a lot to make this video today and make this project successful. I also acknowledge the contribution of the Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery Technology Development Incorporation, both of these institutions located in Mississauga, Ontario, Canada. Thank you very much for watching the video and staying with me. I hope I may see you next time in the same event, because I'm very ambitious. The MES College Marampally may organize the conference on regular basis. Thank you very much. See you soon. Take care. Bye for now.